The next two talks are on medicine, and the first talk is by Professor Robert Hoehndorf, and he will be talking on machine learning with ontologies in biomedicine. Professor Robert Hoehndorf is an associate professor in computer science at King Abdullah Institute of Science and Technology, KAUST. Prior to joining KAUST, Robert has research position in Aberystwyth, am I correct with the pronunciation?
Aberystwyth.
Okay, sorry. Yeah, the University of Cambridge and the European Bioinformatics Institute and the Max Planck Institute for Evolutionary Anthropology. His research focuses on development and applications of knowledge-based algorithms in biology and biomedicine, with main applications in understanding the molecular basis of disease. Over to you, Robert.
Thank you so much for the introduction, and thank you for the invitation to this symposium. So far, I think it was very exciting. I will talk today about machine learning with ontologies in biomedicine. And my talk will essentially have two parts. I'll talk a bit about some algorithms that we built in order to enable this, and then I'll show you how to use these algorithms to gain some insight in disease that we would not otherwise be able to gain. So let's start and just look at the kind of data that we collect in biomedicine now. So we get across multiple different levels of granularity, we can collect massive amounts of data. And this ranges from the genome to the proteome and even the metabolome. And then we have clinical data. And so you know this kind of data, or you will see different types of it whenever you go to a talk about data-driven research in biomedicine or even in biology. This is getting even more as we're getting technologies which can resolve most of these data types, not only on the level of one sample or one organism, but on the level of a single cell. And the big challenge when we can collect this type of data is how can we combine it in a meaningful way so that we can understand how information from the genome relates to the epigenome, to the transcriptome, proteome, and so on. Both on the general data type and then also on specific applications, such as, for example, for single cell sequencing and so on. And in order to enable this, you can see these little arrows here that I drew there. And this is a kind of information which is not entirely data-based anymore. So here we can measure things, but what we'll get is these little arrows. They come from knowledge, and this knowledge is stored in knowledge bases, or they form some micro theories. So you may think of a micro theory in biomedicine as a biological pathway, basically a small representation of how a certain biological process functions and what mechanism is underlying a certain outcome. We can represent this in knowledge bases or knowledge graphs, or we can represent it also quantitatively in some kind of models in systems biology. But the key point is that most of this information comes from experiments. So a lot of this information will come from experiments where maybe a biology or biomedicine PhD student or a postdoc spent a year or 2 or 3 years in the laboratory, and then in the end wrote one paper, and then it becomes one fact. So something like bindings between proteins, the function of a particular gene or gene product, and so on. So this is why we have in biology and in biomedicine very large knowledge bases. They come essentially from over 100 years of experiments in life science. And this is one part of the knowledge that we have available and that we need to utilize if we want to build models in biomedicine which do anything reasonably complex. And there is a second type of knowledge that I will focus on, and these are the ontologies. And ontologies are not things which come from experiments, rather they define our terminology. So an ontology, by the classical definition by Gruber, is a conceptualization or an explicit conceptualization of a domain. But essentially what ontologies do is they describe the classes or the concepts that we have in a domain and how they relate to each other. And they do that in the form of axioms. And I just put two axioms here. These are not things which could be tested or validated by any experiment. They are not the result of any observation. Rather, they are a formal representation, these axioms, of part of the language that we use in biology. So here, a macrophage apoptosis is simply an apoptosis which occurs in a macrophage. And this is a real axiom from an ontology called the gene ontology. I'll say more about this. And then we can go to another ontology called macrophage, which describes what a macrophage is. It's from the cell ontology. It tells us, well, a macrophage, which is the same concept as in the first axiom, is capable of some phagocytosis. And from there, we can then reason about what other processes macrophage apoptosis as a biological process can influence. Now, none of this is testable. It's simply the language that we use in order to describe or characterize our experiments. But how these concepts are related, this is essentially a construct that maybe that a lot of experts have created in literally hundreds of years of manual work in creating biomedical ontologies. It was a very large effort for maybe the past 20 years, since about 2001, when the gene ontology first came out. And these days, there are over 500 ontologies which are used, and they're used in almost every database in biology and in biomedicine. So almost all of them, whether they start from some very basic biological processes or they go to clinical applications, use ontologies in one form or another. So my research tries to find ways in which we can combine the data that we measure, so the original data that we measure, so genotypes, transcriptomes, proteomes, metabolomes in biology, and combine it with what we know to be true, either from formalized knowledge bases or knowledge graphs, they're on the left-hand side of this figure, or ontologies. And then on the right-hand side, you have the data. It's essentially unstructured. And they use very different principles in artificial intelligence. So on the left-hand side, we mainly have symbolic representations. So these are representations which rely on symbols that we can interpret, that we can create, and that we can combine into new symbols, and over which we can reason. So for example, a simple task that we can do on the left-hand side with respect to ontologies and the knowledge that they represent is to ask whether they are formally consistent, so whether they contain contradictions. On the right-hand side, the data is mainly structured. Most of what we are interested in here are recognizing patterns, or to recognize certain regularities, which we can generalize, for example, to predict a certain outcome, a disease outcome, or the trajectory of a biological process. And this is where machine learning mainly is applied. But as I mentioned, we will need to use these connections, or in many situations, we will need to use the connections, so what we know to be true a priori, or based on some experiments, so what we find in our knowledge bases, we have to use this in order to build complex models which can exploit these relations. And for this, we generally use a form of embedding, where we take our symbolic systems, our symbolic representations, and represent them in a space where we can combine it with our data, or where we can add our data in some natural way. And the second part is then the regularities that we want to find in our data, so the outcomes or the relations that we want to identify in our data, maybe a relation between a gene and the disease, or we want to diagnose an individual based on some clinical history together with some image data. We would like to represent this quite often symbolically, because First of all, it gives us interpretability of these results, so we can understand the prediction. We can reason over it, which means we can situate it against what we know to be true already. So we can try to trace back, and we can test whether it's, yes, so we can test whether it's consistent with what we know and whether it's consistent with other predictions that our system may make. And I will start first by talking a bit about these embeddings, because this is quite interesting and goes beyond some forms of embeddings that you may know of, which are maybe knowledge graph embeddings. But so let's try to look at what I mean when I say embedding here, because it is a slightly different meaning than is often used in the machine learning community. So I take the classical meaning of an embedding, where we want to find a morphism or some map from one mathematical structure into another structure, such that the first structure is preserved in the second. Of course, maybe under some different operations. But we want to preserve some properties of the first structure in the second. And here, I mean, the idea of embedding, and I think many of you will be familiar with that, is that maybe the second structure allows us to do some things better, or maybe it enables entirely new things that we could not have done in the first structure, while still preserving some of the properties of the first structure. So for example, our first structure usually will be some symbolic structure, X, that we want to represent in usually some kind of real valued vector space, so that we can use it with modern machine learning methods. So, or we can just use it to determine similarity and so on. And we basically looked into the specific question of embedding description logic ontologies in, so these are particular forms, particular ontologies that use a particular formalism, and they subsume knowledge graphs. So knowledge graphs will be A boxes. I will show this shortly in this formalism, but they go beyond just graph-based representations, and we want to embed them in some RM. And we follow three approaches. I'm only going to briefly mention the first one and then talk about the model-theoretic approach in a bit more detail, because it's quite interesting to look at it, and then show you how to use them. So when I say we use graph-based approaches, they're very simple and they're quite popular, and they are very powerful as well. But the first question is, when I talk of ontologies, then I talk about axioms in a description logic. So you can see them here on the left-hand side. And the first thing that I have to do is convert them in some form of a graph structure. So I need to have some mapping of axioms into a graph. And of course, you can define, or we can define some patterns where we basically take certain syntactic patterns, axiom patterns, and we represent them as a particular type of labeled edge in the graph. So here we have X subclass Y, then we have an is-er edge from X to Y, and so on. So there's a little bit more tricky when we have equivalence or disjointness, but overall, this is something that we can do. Since we use ontologies where we can reason, this is not only something that we can do on the axioms that we assert, but we can also do this on the axioms that we can infer deductively. So I just put three axioms here. So X is a part of some Y, Y is a part of some Z, and part of is a transitive relation. From this, we can, so this entails that X is a part of some Z. So this is simple transitive inference based on these three axioms. And we can do this, this kind of mapping from axioms into a graph, either just syntactically in the asserted axioms, or we can use some kind of theorem prover or some automated reasoning system in order to do this in the deductive closure of the ontology. And then once we have a graph, we can represent this like this. So this is essentially a graph which has some proteins. It's largely what you probably will called a knowledge graph. I put this inferred edge here. So this is just an inferred has function edge between a protein FOXP2 and this gene ontology function, this go colon and with some index. These are classes from the gene ontology. When we have a graph like this, so this is basically, we can do this based on a mapping, based on our axioms, and then we can represent our graph like this, our ontology like this. And we get this graph, and then we can use a number of different forms of embedding this graph in some Rn. For example, we can use iterated random walks, so something like node to back or deep walk, where we generate some random walks, we repeat this, and then use some language model or like word to back in order to embed this in Rn. There are some other group of methods. These are these translating embedding methods. So they have an explicit objective where they have an explicit objective functions. It's based on some kind of analogy based on relations. We would essentially have within some vector space, we basically have that FOXP2 plus binds has to become more or less equal to MET. In this case, if we have this FOXP2 binds MET edge. So there are a number of different ways of embedding these graphs, and then we can use these embeddings. We can maybe use them for some clustering, because then we just use some similarity measure in Rn, like cosine similarity or some other similarity measure, in order to compare them. Or we can just use them as an input to some neural network model or some other model, in order to predict, for example, edges, or do some kind of graph completion, or infer some edges which would likely be true given all the facts in this graph. And we did this quite some time ago, so 2017, we tested this. We just started with a very basic graph where we try to predict the relations. So essentially, we took this graph on the top where we have drugs which target certain genes or proteins. We have drugs which have side effects and they treat certain diseases. Diseases have phenotypes or symptoms. Genes are associated with certain phenotypes. Usually when there is a mutation in this gene, then somebody has maybe a disease with these phenotypes, and genes have certain functions. So it's a very basic, and genes cause certain diseases, yes. So we represented this, and you can see, fine, we can then, we remove some of these edges, then we train some machine learning model, try to predict these edges. This works very well in some of these tasks. So the interesting part is we can do this very generically. Each of these is its own task in bioinformatics. So there are hundreds of papers written about each of these interactions. So in some of these, we could basically get state-of-the-art performance at the time for some of these tasks. But I want to point to something else, because you can see that I report here the performance of this model without including the deductive closure, so without any form of symbolic inference, and with symbolic inference. The idea is that we would either deductively close this graph, or we would not. We would simply just generate our graph based on asserted axioms. And the second one, with reasoning, means we use these inference rules, probe the deductive closure, and then we create a more complete graph. And what I had really hoped to see at the time was that, well, if we add symbolic inference to this task, then, well, our performance will improve, because obviously we have some additional information which is probably very difficult to exploit by our machine learning model alone, and it will not be easily represented in the embeddings. Turns out that this is not the case. So you'll see that there's almost no difference, sometimes a little bit better, sometimes a little bit worse, but there's no significant difference between using this with or without reason. And this was a little bit frustrating to me. So we went back to the drawing board and looked at some different ways of doing this better, because it was my feeling that we should be able to utilize, if we are able to utilize these axioms in the ontologies and in this graph, in the knowledge bases, well enough, then the performance should improve substantially. And when we look at this, if we look at some of these axioms, there's one thing that you'll also notice, that these axioms don't really follow the patterns that I have shown, and they're quite hard to represent in a graph. So these kind of axioms, and you'll also find them in biomedical ontologies. So you have things like cell growth, which is a growth which results in the growth of a cell. And this is a rather complex axiom, so it's very difficult to represent it as a graph in the first place. So this is the second reason of why we need something better. So the question is, can we somehow find a way to embed these axioms directly? So we went and we built some other methods. They worked for some class of problems. They worked quite a lot better. And there also the deductive inference or the reasoning part helped. These were Ontologies and Opietovec. They are the idea, because I'm not going to say much about them. The idea of these methods is that we simply apply a language model on the axioms themselves, and then we can combine it with various forms of other information. So, but none of these, if you use some kind of common language model, then there is no way in which we can make use of the a priori semantics that we know these axioms are supposed to have. And for this, I need to explain what I mean by this. So when we look at semantics or semantic structures, then we need to talk about sigma structures or sigma algebras or interpretations. So we have, usually this is a standard form and it's for a particular type of description logic called EL. This is the syntax and the semantics. So we have syntax, and on top, on the first one is the top concept, and on the right-hand side we have a domain, which is some non-empty set over which we generate some interpretation. And then we have nominals, they're interpreted as singleton sets, and so on. So this is the standard way in which we define the relation between syntax and semantics in first-order logic or fragments of first-order logic. And our idea was then, if we have an ontology with a signature sigma, so a signature here is just the symbols that we use. So we have the symbols that we use, the relations and classes that we have in our ontology. Then our idea was to find an embedding which maps this into this sigma structure, so into an interpretation. So we want to find an embedding which actually generates a model of the axioms in the ontology, and the model means that all the axioms in the ontology will actually be true in this model. So basically this relation is satisfied for all the axioms that we have in the ontology. And we know that this is that we can, at least in theory, do that, because we know that at least this logic, every theory has an infinite model. So any consistent theory has an infinite model. And if it has an infinite model, we also know that we have a model in Rn. So this is just the theorem of Löwenheim's golem in the upward direction. So what we have done is we basically define an embedding for relations, embedding for classes. The idea here is that we map our classes to n-balls, so basically kind of spherical objects in Rn, such that the points which are within this sphere or within this ball will be in the extension of this concept. So each concept is mapped to this n-ball, and then we map relations as a transformation between them. Then we use the axioms of our theory as constraints to build this, and we have some preliminary. So just in the interest of time, I'll just skip through this a little bit. So we can eliminate our A box, so we can focus on a set of normal forms, a standard way to reduce this, and then minimize our loss. We have one loss function for each of these normal forms that we end up with. So just to show this here, so we have essentially, just show it on the next slide. So essentially here for C subclass D, we force C to lie within D. For C subclass of exists RD, then we force C to lie in D transformed by R. So this arrow here in the middle is a transformation by a relation. And when we look at this, you can look at how this generates. This is an embedding of this small knowledge base on the right-hand side. So you can see that here there is male. It will essentially terminate even with a loss of 0 when male lies within person based on the first axiom, and so on. And the very interesting thing is that we are also able to prove a kind of soundness, which means that if we are able to reduce this loss to 0, then we are guaranteed that this theory has a model. So it's consistent. So this is nice because we have some form of formal guarantee, but there are a number of problems. So essentially, and this is ongoing work that we're doing now. So the problem is that intersections of two balls is not a ball. So we switch to rectangles or axis parallel rectangles instead. Our relation model is very simple and the expressivity is rather limited. And soundness is guaranteed, but completeness, so the other side is not. So this is still a big limitation. So we also have a library called MAUL. You can find it on GitHub. We want to make it a hub where we implement all the methods for machine learning with ontologies, with OWL ontologies. So we call it MAUL, and you can look at it. You currently have to look at the develop branch if you want to find some useful information and algorithms, but all the algorithms that are in here and quite a few more are implemented in this library. So this is fine. So this is the part for the embedding. So let's look at what we can do with this. So I start with, I'll talk about two stories here. One is about rare disease. And when we want to, so a rare disease is essentially a disease which affects very few people. They are very difficult to diagnose. Many of them, or most of them, are due to some genetic variants or genomic variants, which are inherited in a Mendelian fashion. Quite often, these are single nucleotide variants. So there's a single base pair in a genome, which is somehow different, and it causes sometimes very tragic disease. Of course, this is not only for diseases. We don't only have to, we are not only interested in diseases. Sometimes it's beneficial if we want to look at a positive trait, for example, from some crop or some relative of a crop, and want to say, okay, somehow we found this wild tomato plant and it has huge tomatoes. So why does it have these large tomatoes? So we want to find out which of these variants, Mendelian here, for Mendelian traits, is related to a certain phenotype that we observe. And there are a number of questions that we have to answer that we cannot easily get from clinical information. We cannot get it from whole genome sequencing data. So we have to know, first we have to find out whether this variant affects a gene function. So for this, usually we use some kind of structural methods. We have to find out which gene. For this, we need to know the annotations of the genome, of the human genome, where is which gene and how do variants affect particular genes. Then we need to understand the pathways and networks in which this gene functions, or this gene product, usually protein, functions, in order to exhibit, or to do what it does. Later on, we need to understand how this affects the physiology of the organism, and if there are any interactions with environment, then this is also quite challenging. So we need to understand all of this. And most of this information, or a lot of this information, can be found in both knowledge bases and in biomedical knowledge bases and ontologies. And for this, we have built, so we need to rely on some background knowledge, and this is something that we find in ontologies and knowledge bases. So we have built a system, and it's essentially, I just show these results first. So it's essentially a system which takes a phenotype, which is essentially the signs and symptoms of an individual with a rare disease, together with whole exome, whole genome, or some kind of genomic information, so sequencing data from this individual, and tries to find out, or determines which variant causes this phenotype. And there are a number of tools. So ours is called PVP, for phenotype variant predictor, I think. And so we have various forms of this. And you can see that this works quite well, better than related methods, mainly due to our ability to utilize more background knowledge, biological and medical background knowledge, in the form of ontologies. And I'll show you, so this is for single nucleotide variants, but I'll show you how this works on a bit more complex example, and this this is related to structural variants. It's more interesting to looking, and most of this is also true for single nucleotide variants. So structural variants are not changes of a single nucleotide, but they are changes of usually quite a lot of nucleotides. There might be a whole arm of a chromosome maybe missing, or large parts of a chromosome are duplicated, and there's quite a large amount of structural variation in the human genome, and there are many associations with disease. So many of the structural variants have the possibility to cause a set of abnormal phenotypes. But the problem is that now we are not even dealing with a single gene, like a single nucleotide variant would normally affect only a single gene. Now we are dealing with things which may affect hundreds of genes if we are unfortunate. So we need to add some additional models. So we have built a system called Deep SVP. I'll briefly describe how this works. So it takes two inputs. It takes as inputs phenotypes. So this is clinical information. So it's essentially what are the symptoms of the disease in one individual, and these are rare diseases. Many of these diseases don't have a name, so we cannot just say, okay, it's a disease. We have to break it down into its symptoms. So these are the symptoms, signs and symptoms, and then this VCF file. This is the genomic information. So we do the first thing. We essentially use an embedding method which combines ontologies, quite an amount of background information, including protein-protein interactions and so on. I have this on the next slide, and generates an embedding. Then we have an embedding for each gene based on what we know this gene is doing, the functions of this gene, the phenotypes, which if we know any phenotypes associated with this gene on one side, and then either the disease or the individual patient phenotypes will be the second kind of embedding. And then we have some model here which will predict for each gene and for each disease whether they are likely, so whether this gene is likely causing or involved in this disease given only by its set of phenotypes. So this disease is not a disease in the normal sense. It's basically just specified by its symptoms. And this is important because, well, we don't have the disease name. We want to find this out. So the second part is, so this is basically standard. We have some annotations, different type of features. So how many genes are affected by the structural variant? So how long is it? In what region of the genome does it lie? So is it maybe something that is very common in a population, or is it something that we have never seen before? So this kind of set of features we generate here. And then there's an aggregation function. And this aggregation function here takes possibly multiple genes, and then needs to find out how this might relate to this structural variant. And we use two aggregation functions, and they correspond to two different modes in which this structural variant would cause phenotypes. One is a maximum, simple max function, which means that there's a single gene in this large variant, which maybe having affecting 100 genes, but there's only a single gene which causes most of the phenotypes that we see here in this individual. And we use an average. We can also use a top K, but we use an average as a second way to aggregate, where we assume that somehow all of them contribute, maybe different, they have different contributions. And then this goes into a machine learning model, all combined, and it predicts for a structural variant here, a copy number variant. It predicts whether it's likely to cause the set of symptoms that we have seen in the individual or not. So when we, there is a second issue, and this is that we have very little information about genes. So by using these embeddings, we essentially implicitly learn to predict gene to phenotype associations. And we learn to do that from the functions of gene products, the anatomical side of gene expression. So where in the body is this expressed, the cell types in which this gene is expressed, and some information from model organisms. And this is all integrated, then we can embed this and use this. So when we look at this, this works way better. So the bottom three are our baselines here. So this is essentially what we would use if we had not done this. But you can see that this is that this works quite well. This works very well. There are different ways. So this is basically average and maximum aggregations. Then we test this for different ontologies. So this is basically doing this only based on gene functions, mouse phenotypes, human phenotypes, cell type, anatomical site of expression. And here the union means we combine all of this together. So you can see that we can improve substantially by making use of background information, not only in knowledge bases, but also in the ontologies. So this is something that is very nice. And okay, so I have not much. I have one more point that I want to make. So I want to talk about a second application of our embeddings. And this is predicting drug target interactions. And it's very interesting. This is very recent work. It was just published a few months ago. And we call it, you see by the title, it's called Drug Target Interaction Voodoo, because I'm going to talk not so much about how nice our model is and how challenging it was, but rather I want to talk about some biases that we can find by using also ontologies, only briefly. Because when we predict drug target interactions, this is not a single task. So we have to answer first the question of what is it that we actually predict here. So one formulation would be, given a set of drugs and proteins, so we have two sets, some drugs, some proteins, we just want to find out of all of them which drug interacts with which protein. This is a very global perspective, and we wouldn't actually care much if all the interactions are with a single drug. So maybe one drug interacts with every single protein, it's very prolific, then we would always be right just by predicting a single drug. You'll see that it's maybe not the most useful perspective to take. So we can also ask the question, given a protein, what are drugs which potentially target this? This is a protein or disease-centric perspective, because if we know this protein is somehow involved in a disease, just as I have shown, maybe even with copy number variants or structural variants or single nucleotide variants, maybe we want to inhibit this particular protein. So we are looking for a drug which inhibits this. And we can do this the other way around. Also, we can do this for, given a drug, we want to find the proteins, then this is maybe a way in which pharma companies would find new uses for that drug. But the key point is that the evaluation and probably also the training will be quite different. While the global perspective, you would simply take all the predictions of all the pairs, generate some macro averages just over all of them together, all in a big list, and then you can kind of report performance. If you do this per protein, then essentially you have a specific classifier for each protein. So you need to compute and report micro averages. And same thing, you will probably have to split differently because the dependencies that you assume your training and testing data to have will be different. So in the global perspective, it's fine if there are certain dependencies between training and testing data, because that is kind of what this model is supposed to exploit. While if you have given a protein which drug targets this, you would probably want to split by proteins so that you can determine how this actually works if you have never seen this protein. So we have built this model. It's quite interesting. It has two sides. It generates some protein embedding and drug embedding. It uses our ontology-based embeddings together with some structure embedding, so based on protein sequence. Same for the drug. It uses some ontology embedding, uses different type of information about drug side effects and so on, together with a structure embedding of the drug. So the chemical structure combines this. We have a graph neural network that we use with some residual connections, mainly the graph is a protein-protein interaction network. So it's essentially, the idea is that the effects of drugs will be the result of a network effect in some cases. So we wanted to have this as well. So, but let's look at this. Okay, I only have 2 minutes left. So let's look at the results of this. What we'll find is, usually you get people to report in bold, they always do the best. I'm not doing this. So we do this, then if we do this macro ARC, Voodoo does not do this. It does not actually outperform any of our state benchmark methods. And this naive predictor, which I will, so this naive predictor is something that we have built. And all this naive predictor does is it ranks for a protein, it simply ranks every drug based on the total number of interactions that are known for this drug. So for every protein, it ranks, drugs are ranked exactly in the same order, just by the total number of interactions that this drug has. That gets close to state-of-the-art performance, even in macro ARC. And we're even worse than this if we do this. But if we look at micro averages per protein here, you can see, so in a protein split, and we split by protein, then you can see that Voodoo, or our Voodoo method, actually works much, much better. And you see also that this is very consistent because of the way that we have trained in the different evaluations. And this was exactly the aim here, to have this balance and to get rid of these biases. So, okay, I think my time is up, so I'm going to not talk about this. There are a number of biases that you can find in drug-target interaction prediction, and it's not a single task, it's multiple tasks. They're quite often conflated. If you want to know more, you can look at our paper. It's just out in Bioinformatics on this. So I'm going to skip this and we'll conclude. What I'm trying to say here, or what I was trying to show, is that the domain-specific knowledge is something that we need to utilize to build AI models that are robust and that will work in or will be able to solve complex problems. And this is not my idea, so this goes back to a very long time, Feigenbaum. He proposed this for search in discrete spaces. But essentially, I think this theme that in the knowledge is the power is as current now as it was in 1977, only that the type of search has changed from search in discrete spaces to search in continuous spaces or in vector spaces through optimization algorithms. And the embedding methods that I have shown and some of the applications where I've shown you how they work will be able to make some domain-specific knowledge available to machine learning models. I want to thank my group and thank you very much.
Thanks, Robert. That's a great talk. And I see the number of questions are also too many. So let us start. Shomak, you want to go?
Sure. Yeah. Thank you, Robert. Fantastic talk. And I really like the fact that you have succeeded in integrating this type of structured knowledge in bioinformatics. I have a specific question. Can you please repeat, how did you compute soundness? Because based on that existential, I think, language, EL language, right? Existential language, even there is existential quantification. So even there, I think you will need a lot of approximations, right? Even without universal quantification. So I would really be interested in approximations.
Yes, this is a great question, because I kind of abuse a little bit the notion of soundness here. And I also have to say that the result is to a certain degree trivial. So soundness here, by soundness, I specifically mean that if we are able to reduce the loss to zero, so if we can solve this optimization problem, then our theory is guaranteed to have a model. That is, of course, true. So without the other direction, the trivial way in which this can be true is if we can never reduce our loss to zero. So if our loss is guaranteed to always be higher than zero, then this proof is trivially true. So there are some additional assumptions. There are at least some non-trivial cases where this actually works. This is not one. But I think you're referring to this kind of existential restriction. What we are trying to do here is that we are trying to embed the axiom, so this is C subclass exists RD, in such a way that all the points within the space that lie within these circles, within these balls are classes, so that lie within these circles lie in the interpretation of R. So here our interpretation of R is a certain transformation. This is basically it's basically that if we add R to any of the elements in D, so two elements stand in relation R if one element from X and Y stand in relation R if X plus R equals Y. And that is the formal representation. So it's basically just this shift by a vector called R. And this is essentially just the trans E property. And what this loss does, I don't write the loss here, but what this loss aims to achieve is that this remains true for every element in the extension of both C and D. So that if we apply C plus R, it will have to lie within D.
Sure. Yeah, great.
Right. Makes
There's
sense.
another question. Abhishek. Abhishek, you want to ask a question? Abhishek? Okay. So there are one more question. Let us take one more question. So can embedding be used for detecting aggregation prone regions in protein aggregation models? Are there any embedding softwares available? So this is from the.
Just a second, I'm not sure I get the question. So
It's in the QA, the last question. I'm taking the last question. Can embedding be used for detecting aggregation prone regions in protein aggregation models?
I am not sure. I do not know. To be honest, I do not know. I do not know enough about protein aggregation models to answer this. Maybe I can get back to you on this question.
Great. So thank you for this talk. It's a great talk, and this topic is also very, very interesting and upcoming. So there are some questions which I have not read out. So if you can read out and answer them during offline, that would be great.
Thank you very much.
Thank you.